Good morning, everybody. Welcome to the No-Till Farmer webinar. What's the big deal about biologicals? Focusing on biointrinsic by Indigo. Today we have 4 guests joining us. We're very fortunate for their attendance. We have Mekdem Tesfaye, who's worked for Indigo Ag for about 5 years with the primary focus on microbial isolations. She's going to be talking a little bit about how the company identifies soil microbes and how they go about building a library of various soil microbes to help grow crops. Kevin Calzia is the Senior Director of Formulations and Quality Assurance at Indigo Ag. He's also worked for the company for 5 years. He's going to focus a little bit on how it gets from their microbial library to the finished product. And then we're also joined by Matt Powell, a certified crop advisor and technical services manager for Indigo Ag. He's based in Iola, Kansas. He's also been in production agriculture for a number of years. And Matt and PR Morris will be jointly presenting. PR is another certified crop advisor for Indigo and another 5-year veteran. He's based in Lexington, Tennessee. He's been working with production agriculture for about 13 years. Before I turn things over to our panelists, I just have a couple housekeeping items to take care of. On your screen, there is a control panel with a Q&A icon that will allow you to submit questions at any time during the presentation. And we'll try to get those questions answered at the end of the session. Well, we do ask that you enter those into the Q&A and not the chat, so they'll all be in one place. If for any reason the webinar is disrupted or you're disconnected or seemingly disconnected, just go back to the invitation email that you received and rejoin the webinar through that link. With that said, thanks for joining us, and I'll turn things over to Mekdem.
Hi, thank you for the introductions. And before I move on, I just have to make a slight correction. Our isolates come from inside the plants or from the rhizosphere. It's not necessarily the soil. So I'm pretty excited to be able to speak to a very important group of people, farmers, and they're important to what we do every day. And I will now discuss the discovery process we follow to identify and source the agriculturally beneficial microorganisms, which we call our focus sourcing effort. So the underlying principle of this strategy is from plants for plants approach. So the explosion of research into the human microbiome over the last 20 years have revealed that there are as many microorganisms in and on the human body as there are cells. So recently, this research is being paralleled in agriculture, and this is known as the plant microbiome. These microorganisms live on the inside and or near the surface of plants. These microbes have co-evolved with the plant and are also as important to the health and development of plants. So Indigo mostly focuses on a subset of these microorganisms, mostly endophytes. These microbes make their homes inside plant tissues, forming a close long-term association with their plant host. So on the right-hand side here is an image, it's a microscopy image of one of our advanced commercial strains, which has colonized the interior of a root hair. This wheat seed has been treated with the strain and has been tagged with a green fluorescence protein. That's why you're able to see it all glowing. So what you see here in the red surrounding it is the root surface of the lateral roots, and the green dots are the strains that have colonized the inside of the roots. So what are microbes doing in plants anyway? What we are learning from our microbial library at Indigo and what microbial ecologists have known for a long time is that there's probably a microorganism that has evolved a mechanism to combat or mitigate a variety of environmental stresses. That includes both biotic stresses listed here on the left-hand side, like a fungal disease or insect attack, or abiotic stresses like nutrient deficiency or drought stress, which is listed on the right-hand side. So I'll give you some examples here. So some of the mechanisms by which microbes can improve plant health under stress. Beneficial fungi often produce antibiotic molecules that inhibit the growth of pathogenic strains. So on the bottom left here is a fungal strain plated on 4 locations around the outer edge of the plate, which is inhibiting the growth of the Rhizoctonia pathogen that was plated at the center of the Petri dish. So for biotic stress, for abiotic stress, some bacteria produce plant hormones that can improve plant growth or alter plant physiology. The image here on the top right panel shows a plant colonized by a phylobacterium that produces a plant auxin. The bacterium causes a dramatic increase in lateral root formation compared to the untreated on the left-hand side, which may help the plant take up water or nutrients from the soil. So we seek to understand what our microorganisms should be present, what microorganisms should be present in a healthy plant, and then reintroduce those missing plants and environments through our microbial seed treatments. So you can think of our seed treatments as a sort of plant probiotic. So the following 2 slides gives an overview of our focus sourcing strategy. We start from the complexity of a microbial community consisting of tens of thousands of unique strains of bacteria and fungi that is found in just 1 gram of soil. We first use the plant as a filter. As mentioned earlier, indigo has made the focus on mostly endophytes, which harness millions of years of plant microbe interactions to filter for those microorganisms that form intimate relationship with the plant host. Then we take that 1 step further and try to understand what microbial community in a healthy plant looks like. We travel to agricultural fields, experiencing a stress of interest, and find the plant that seem resilient to that stress. We collect those healthy plants and their non-healthy counterparts, and we sequence all of the microbial community present in these 2 subpopulations. Is the microbiome of the healthy plant that's giving it an advantage, and sometimes we find out that it is. So I'll give you a real life example where a field in Iowa was experiencing a stress, and focus sourcing took a trip to Iowa, and this particular field was showing symptoms of nitrogen deficiency, and this was confirmed using tissue and soil nutrient analysis. The team traveled to Iowa, met with agronomists, and collected a cohort of healthy plants that were showing some resistance to the nitrogen stress, and a cohort of plants that were showing obvious symptoms of nitrogen deficiency chlorosis. We brought those subsamples of rootstock and leaf tissue from dozens of plants back into the Boston labs, sterilized the surface of the tissue, and sequenced the microbial communities. That's both fungal and bacterial communities associated with the healthy plants, and also the bacteria and fungal communities that were associated with the chlorotic plants. So the microbe sequence confirmed that there was a large difference in microbial communities. The winner communities are clustered in the red dots versus control communities, which are clustered in the green dots. On the right panel, when you dig into the individual microbial taxa, what is driving these differences in community structure? Are the 2 ASVs that were specifically enriched in the winner plants versus in the control plants or the unhealthy plants. And after further investigation, it showed that these two microbial groups are comprised of known nitrogen fixing organisms, supporting the hypothesis that the microbes associated with the healthy plants are providing them an advantage in this stressful environment. Aggregate data across breadth of our sample library allows us to see, using a sequencing approach and the collection effort from healthy versus chlorotic plants, we have collected over 8,000 plant tissue samples from across the US from various crops and various environmental conditions. For each of those tissue samples, we have detailed sequences, record of all the microorganisms that reside in each of these tissue samples, giving us a deep understanding of the plant microbiome communities. So doing this, we have built out the most diverse library of endophytes in the world. We have over 25,000 crop-associated microbes isolated and identified to date. Of those, 4,000 are distinct genera, and hundreds of those are noble species. Of these, we have filamentous fungi, yeast, bacteria, and some algae. And we use this data, we isolate and identify and sequence the genome of each of these unique microbes, allowing us to predict or nominate which strains might be best candidates for testing in our R&D pipeline. And I think Kevin is next. And also we'll take your questions at the end.
Yes, so hello all, my name's Kevin Calzia. I lead the formulations and quality assurance group here at Indigo. And as Magdam just described to you, all these microbes that are isolated and identified through the focus sourcing program are then evaluated in laboratory testing and then greenhouse testing and ultimately field testing. And as they go through all this individual testing protocols, at the end there, we actually have to make a product out of these microbes so that they can actually be used and delivered to the retailers and the farmers and provide their benefits back into the field. I should note that at this time, Indigo really just focuses on seed treatments, though we have talked about other methods of application for our microbes. All of our microbes are developed to be applied as seed treatments. So one of the biggest challenges in delivering microbes as seed treatments back to the field is making sure that those microbes stay alive and can actually colonize and interact with the plant at that time of planting. So much of the development efforts in my group and groups supporting me are around stabilization and keeping that microbe alive from the time that it actually goes into the package and goes through the supply chain and delivery to retailers or to the farms. But then also they have to survive through the seed treatment process and then ultimately stay on the seed for a period of time until planting. And then once planted, since those microbes are still alive, they can then grow with the plant or with the seedling and actually colonize and provide the benefit in the field. So the way we do this is by keeping the microbe dormant. And when I say dormant, I mean the microbe, as I said, is still alive, but is really in a sleepy type of state. So it's not actively growing in that package, and it doesn't grow until it's actually put back into the soil where you actually have nutrients and water, and then that microbe will start to grow, much for the same reasons that as you put the seed in the soil, it starts to grow because now there are nutrients and moisture around to allow that seed to germinate and grow. So we achieve that dormancy all through we aim to achieve that dormancy all through the development cycle. And that's what I have shown on this slide here. So at the very beginning, we have to figure out actually how to grow these microbes industrially, at large scale and at high concentrations. So we do that through fermentation. The fermentation processes are akin to what you might think for like brewing beer or other sorts of industrial microbial growths. And in addition to just achieving high concentrations, high quantities of these microbes, we also target very specific physiologies. And you can think of this in the sense of you don't want to have a microbe coming out of fermentation that is already on its way out to being dead, right? We don't want an old microbe coming out of the fermentation. We want one coming out of the fermentation young and healthy. So we do that by adjusting the nutrients during that fermentation, the conditions of that fermentation with mixing different dissolved gases. And so that microbe comes out feeling nice and young and strong. And that already gives it a heads up in being able to be stable and stay alive through that time period to planting. Some microbes coming out of fermentation need a little bit of additional help, and they go through some downstream processing steps. So in those steps, sometimes we include various additives, and we introduce other processes too to change the microbe from being in a liquid state to maybe being in a powder state. And you can think of that too, similar to like food packaging, where some foods are okay and they can stay preserved for a period of time in a liquid state, but other foods are more akin to being preserved and shelf stable as a powdered state. So again, depending upon the type of microbe we're working with, we go through fermentation. Sometimes it goes through some downstream processing, but all the microbes ultimately have to be formulated into a final product that has certain physical characteristics as well, not just the biological characteristics of the microbe. So typically these formulation processes are a blending process. We introduce other additives. Maybe there's a bit of a dilution to get it down to proper concentration for dosing on the seed. Sometimes we add components to impart certain flow characteristics on the seed, adhesion to the seed, and things of that sort. So again, go through the fermentation, the downstream processing, the formulation development, and then all those to come together, and we actually have to scale them up for commercial manufacturing. So Indigo works with a variety of contract manufacturers where we take all those laboratory processes and conduct them at large scale, industrial scales, to produce commercial quantities of our products. All those products then go through a rigorous quality assurance check where they're tested against set specifications to make sure that they meet all their requirements before they're actually released as a commercial product to the retailer or to the farmer itself. Indigo's products are available in 3 different formats, and these different formats are one chosen because of the type of market that the product might be sold into and the way that product may be used in that market, but it's also controlled a bit by the type of microbe. And so one type of product that we sell is called a water dispersion. So this is a typical liquid seed treatment. It's applied much like other liquid seed treatments, other chemical pesticides are to the seed. And this tends to be a favorable product type because it's common, it's accepted in agriculture, and it's used with standard seed treatment equipment. The microbes that are supplied as water dispersants tend to be ones that are a little bit more robust and a little bit more stable. The next type of product that Indigo offers is called a wettable powder. And so this is a product that arrives as a powder, and then it's dispersed in liquid at the time of seed treatment and then applied much like any other standard liquid seed treatment. Microbes that go into this type of product format may need a little bit more assistance in terms of the stability. They prefer to be in a powder type format and maybe aren't as stable in a liquid type format, but they can still be applied as a liquid. The third type of product Indigo sells is called a flowable powder. And so this again is a dry powdered product, but it's not dispersed in a liquid. It's actually applied as a dry powder directly to the seed. And this is amenable to the type of microbes that maybe are the least stable, where again, they don't like to be around moisture, and to keep them alive until the time of planting, we keep them dry through that life cycle of them from the packaging to the seed treatment and to the time of planting. So again, multiple product types, depending upon the type of microbe. Each of those product types have different storage requirements, different ways that are applied to the seed, and ultimately, deliver different types of characteristics and benefits to the plant. So as I mentioned with each of those product types, all 3 types of products can be applied, using standard seed treatment equipment at a retailer. So that is one approach that they can all be applied through. Other application methods are more on farm at the time of planting. And those seed treatment methods are either manual or sometimes with an automated sort of powder feeder. Typically, the on-farm treatments are for our flowable powder type products, and those can be added to the seed as it's being transferred or conveyed into the planting equipment itself, or it can even be applied at the individual row unit. One key thing to keep in mind that when our flowable powder products are being applied is that as they put on the seed needs to go through some sort of transfer or mixing type step. So whether that's movement up through an auger or whether that's through more of a manual mixing step with an auger bit on a drill or something in the row unit itself. But it usually takes very little agitation and movement of that seed to evenly disperse our powdered products onto the seed, much like if you were to add a seed lubricant, an 80-20 of some sort onto your seed. So as Indigo has developed these different products for seed treatment, we go through, of course, laboratory development and testing, but we also take them to large-scale pilot trial seed treatments. So the picture you see here on the right is from one of these pilot trials that Indigo has conducted, and we treat seed in a variety of manners to make sure that, one, the treatment is uniformly covering the seed, also to make sure that that treated seed has a corresponding or correct physical characteristics with respect to flowability, dust off, plantability of that seed. Make sure, of course, that the microbe stays viable through these large-scale seed treatments. We confirmed that in laboratory testing and small scale, but we want to make sure that with the large equipment being used, that again, it holds true. And of course, in this large-scale testing, to make sure that the product is easy to use and compatible with all the larger pieces of equipment. So, as mentioned, uniformity and coverage of the seed is of course paramount because we want to make sure that each individual seed is going to have the appropriate dosing of microbe on it so that each plant in the field is going to see the benefit from that microbe. So what one has to keep in mind when applying microbes to the seed is the difference in size scale between the microorganism and the seed itself. And so if you have an indigo powdered product and you follow the instructions and take a half-ounce scoop of that product and apply it to a 50-pound bag of seed, in each little half-ounce scoop that you're applying to a 50-pound bag of seed, there are literally more than 10 billion microbes in that scoop. As that half-ounce scoop and the powder in that scoop is distributed across the seed in there, you now have roughly around 10,000 microbes on each individual seed. And so if we're able to look at a corn kernel there as shown in the upper right, that corn kernel is maybe 10 centimeter in length. If you could zoom in with a microscope on the surface of that corn kernel after it's treated with our microbial products, those microbes are more than 1,000 times smaller than that corn kernel. So you don't really cover the seed with the microbes, but you're just putting little spots of microorganisms onto the seed itself. So we can take each of those individual corn seeds after they're treated in our laboratory, and through specific methods, we can extract the microbes from the surface of that corn seed and then count them. We can quantify them. So the plot you see there in the lower right, that bar chart, that's where we've taken actually individual corn seeds, extracted the microbes from them, and then counted them on what's called a microbiological plating method. You can see that each of those bars is roughly around 10,000 individual microbial colony forming units on each seed. So we have nice uniform coverage, but we're also getting roughly the same dosing as well, 10,000 CFUs per seed. And so this means that you're going to have all the seeds covered, but also the same level of dose and the same level of response for each of the corn seeds that have been treated. So hopefully that provides a little more insight to how we develop our product, take those microorganisms that are identified through our focus sourcing, develop them into products, and then actually build the confidence and confirm that they're going to be viable and cover each of the seeds as the seed is treated. That, I'll hand it over to Matt.
All right, thanks, Kevin. Good morning, everyone. My name is Matt Powell, and I'm an agronomist working as a technical services manager with Indigo. I cover primarily eastern Kansas and eastern Oklahoma, and a big part of my job, as well as PR's job, is we help support the sale and placement of our biotrinsic lineup, as well as placing field trials out there with retailers and growers as well.
So I'm PR Morris, and I am the technical services manager down in Tennessee, covering the entire Southeast, Arkansas, Louisiana, over to the Carolinas. And my expertise is going to be in corn, soybeans, and cotton.
Okay, so I'll go ahead and get us kicked off this morning. We had some farmer questions here that PR and I often get, and just kind of wanted to go over those. But before I do have a confession to make. I was, as an agronomist in the industry, I've been doing this for 25 years now, and I always had a little bit of skepticism around biological products in general. And I know in talking with growers, there's you've had that biological seed salesman that's come up and he's got that bug in a jug or pixie dust that's going to make your worst field the most productive field on your farm. And I've heard that as well. And when I came to Indigo almost 5 years ago, I learned that things have changed considerably. And to Kevin and Mechnam's presentation, I think you can understand the advances we made, both in understanding how biologicals work within the plant, the interactions that we see with them, and then also the delivery and performance of those products on seed once we get them to the field. So we've made some drastic improvements with biological products in the last 5 to 7 years. And so going back to those farmer questions that we often get is, how do I know if a biological is right for my farm? And I would say that there's very few farms that wouldn't benefit from a biological product being applied. We're seeing, with our products, 75 to 80% win rates when we put these in the field. And so, the main thing is identifying what is the challenges you're dealing with. What's that limiting factor or factors that you might be dealing with in your corn, your soybeans, whatever that crop may be. And address that through a microbial. And we have microbials that can help with heat and drought stress, with nutrient utilization. We also have newer products that are also improving with seedling diseases as an example too. So there's a lot of like I said, about every farm could benefit. And I know that the audience here is a lot of no-tillers. You guys have been focused on soil health probably for a number of years, and one thing that biologicals do is they're a means to improve that soil health as you continue to go down that path. They're natural products. They don't have any detrimental impacts on soil health or the biology within the soil. So that's a great thing as well. Pardon me. So what are the key benefits of using biologicals? I guess I always go back to, you want to see a return on that investment, through a yield response or, a response to a stress, an improvement that you would not otherwise see if you didn't apply that biological. The other thing that biologicals are is they're extremely safe. Again, most of them are naturally derived. So, they're safe to the environment. They're safe to apply them, and so that's also a key benefit. And then which one should I try? Well, there again, I would go back to what you're dealing with as far as the stress, and look at the that biological that you feel like can work best for you. One thing that I feel like we do a great job at Indigo is we have a lot of field trials out. We work with land grant universities to put independent trials out, as well as other third-party companies. And so, if you can find data that's local, obviously that's a great thing. And I always try to encourage growers to do that, because with these biological products, I mean they perform differently under different soil conditions, different soil types, different environmental conditions. So there's a lot of variability that goes into how they perform out in the field. So having that local data is really critical before you decide to put one of them out yourself.
So the type of microbes that we have and what we're trying to target with each microbe is either drought tolerance, heat tolerance, nutrient use efficiency, and disease management. And even though I don't know how much irrigation is where each and every one of you are, but it seems to be one thing that has really taken off down south. And even with irrigation, we can have times that we still have drought stress, and we have a lot of heat stress down south. So that's exactly what we're trying to target with these microbes, is trying to help that plant just mitigate that amount of time that it's in that stress. We also have a few microbes that have those nitrogen fixating bacteria that are present, that we can actually mine up some organic nitrogen and make it available for the plant. And in doing that, we also reduce disease pressure and disease, or we manage those diseases a lot
easier. Okay, so I'll hop in here. We're PR and I are going to kind of tag team on what we've seen with different crops in our area, and we're primarily going to focus on one particular product for each crop, just because of the time restraints we have here today. But for each of these crops, we have multiple products, and we kind of place them based on the data that we have and how well they work in a particular environment and soil types, as an example. So. But I'm going to go over corn here and kind of show you what I've seen the last few years. But this product that I'm going to focus on is W12. It was actually commercially new in 2022 for us, and we've seen really great results with it. But with a corn plant, most of you all are familiar with how a corn plant grows, obviously, and what kind of an impact stresses can have once you get into the reproductive phase of corn. And this graph on the right just kind of depicts that. We can see up to 50% yield reduction, if we have stress there, around pollination and grain fill, early grain fill. So, with W12, what we've seen with this product, I mean, it's taken in immediately by the plants through that, through those early roots. And with it being an endophyte, it's taken in and it performs season long throughout the year. So, we see bigger roots, healthier plants, and we'll show some pictures of that here coming up. But that's kind of how the product works within the plant, and how it provides a yield improvement once we do hit those stressful periods. So this is the picture on the right here with the map, just shows some yield improvements we've seen with W12 this year. Like I said, it's performed really well. I just saw an update this morning on this particular product. We've had an 85% win rate in trials, grower trials, and across the U.S. So it's performed very well. And here on the right, I just want to show, this is an example of kind of some of the things we can see out in the field during stressful periods. This picture was taken by our counterpart, Chris Lobmeyer, out near Garden City, Kansas. And you can see on the circle on the left how that plant is stressing, and it's got that blue-green hue. You guys have all probably seen that, when we get it during a stressful period, when the corn water use requirement really starts to increase. And but you look there on the right with the biotrinsic treated picture, and we're still wilting, and we're still seeing some stress, but we're not seeing that visible blue-green hue to that plant like we are in the untreated side there. So this is a retailer trial that we had up in north central Iowa, and this, they say a picture says 1,000 words, right? So this is pretty obvious. I mean, the treated here is on the right side of all of these photos. This looks like about V6, V7 corn, but you can just see the difference in root development, the better above ground growth as well. What's pretty eye-opening to me is the difference in stalk diameter between the treated and the untreated here. And it just it just goes to show that, yeah, this product is working early on, and is really improving early growth out there. And a lot of times what we see is that growth improvement and early health improvement is carried throughout the season. This is a photo that was taken down in PR's area down in the Arkansas Delta, so I'm going to let him speak to this one.
Yeah, this was very eye-opening with the drone, kind of like that picture we saw couple of slides ago where that corn was really rolled up on the left side of that blue-green hue. This corn was about V12. Tassels were going to be coming out within the next 2 weeks, and I sent the drone up and was able to capture this, and it was pretty dramatic. You can see on the left-hand side of the screen that was planted without the treatment, and there's a little hook about midway to that picture there, and that's where we stopped the planter. We filled up the hopper boxes with the powder treatment, the flowable powder, and continued planting the rest of the field. And over on the right is the yield mount, and this corn was harvested in September, and the grower sent me the yield mount, and he said it's pretty dramatic. And once I guess did an analysis on the data, you can see that there's a 29 bushel increase where the treatment was as opposed to the untreated plants in the field.
So this is a case study of one of my growers in southeast Kansas and near Fall River, Kansas specifically. But this was a retailer put this out for us. It was a variety trial, and we had the same corn hybrid, treated and untreated, 6 rows of each right next to each other. And I actually got a call from the retailer on this one, and he's like, the corn was probably V2 when he called me, and he's like, man, I can see a difference, And so I went out a few days later, and sure enough, I mean, we could see it to the line, where the treated had, better early growth, better early root development. Here's some photos, showing kind of some of those things that we were seeing out there. This is probably when the corn was about about V4. So, this corn continued to have that difference throughout the growing season, kind of like I alluded to earlier. Once, we got the corn was chest high, there was probably 6 or 8 inches difference between height of the treated and the untreated. We went to harvest with this one, and this particular plot was under some pretty significant drought stress, especially later on. We didn't get much rain past early June, really. And so we had a yield environment in that 120 to 130 bushel range in the plot. But you can see the bushel uplift here right at 13 bushel per acre. And it was pretty eye-opening. And when I went out and did yield estimates on this, the main thing that I saw was, there wasn't much difference in ear length, but ear girth was different. And that's where we gained the difference in yield. And you all probably know, but corn, you're going to determine that ear girth around V6. And that's kind of when we started seeing some of that heat and drought stress set in early on. And the treated, I was consistently running 18 around about every check I did, but about on the untreated, about every other year was 16 around, so there was a definite difference there. And so, I always like to bring it back to the grower as far as return on investment, because that's what it's all about, right? I mean, that's what, it's all about your bottom dollar and how much are you spending on these products and how much are they giving you back. And so with most of our products, I mean, it's going to vary. With all of our products, it's going to vary depending on seeding rate. But with our corn products, we're right in that $4 to $5 an acre range. And you can see here our profitability gain was, right at $85 an acre, in this example. So a great return on that investment, obviously.
So I'll speak to soybeans here. There's one product that we have for soybeans. So I guess we're doing the keep it as simple as possible here. And that product is M34. We have that in a flowable powder. We also have that in a wettable powder. And in the wettable powder form, we have it with an inoculant, which is M13, which is just rhizobia bacteria. And then it's actually 3 strands of rhizobia bacteria. And then we have an extender that gives us up to 120 days upon seed. So you're not having to treat and then get it in the ground as soon as possible, because we understand issues happen with the weather. So just some of the things we're seeing is a 20% shoot biomass and 154% increase in root biomass. And these pictures here were taken in the lab, and we've got some more from actual infield on a couple of slides later. So this picture is from Western Tennessee, about halfway between Memphis and Nashville. And the picture on the right, the treated is on the left side of that picture, and the untreated is on the right side. And these are the same plants in the left picture. I've just stripped all the leaves off to look at stalk, fruit set, and do a pod count. So it's the same plants, and what we've seen is a bigger root mass on the treated. We're seeing more nodules on those roots. We're also seeing a larger stalk diameter, and we're holding on to more pods. Matt, what did you say you were seeing as an average on pod count?
Yeah, so a couple of years ago, I did we had some stress during pod fill, and did a similar analysis here as what PR did. Basically stripped the plants and counted the pods, counted them and weighed them both, and we were right at a 15% difference in weight and number. So yeah, I mean, like PR said, I mean, we are able to retain those pods better, when we do get in those stressful situations. And also what I've seen is we don't lose beans within the pod when we hit the stress. A lot of times we'll go from a 3-bean pod to a 2-bean pod once we hit stress, but we don't see that as frequently with this N34 product.
So this is a plot from this year that I did. That these are 4-row plots, and over on the picture of the seedlings, you can see that the treated is on the left, those two on the left, and the untreated are those two on the right. And again, we're seeing a larger root mass, more lateral roots coming out, more root hairs, exploring more soil with those treated plants than we are the untreated. And in the picture of the soybeans there on the right, just about middle of that picture is the dividing line of treated and untreated. And the 4 rows to the right are starting to lose leaves. They're dropping leaves up in the upper canopy. And just the treated plants are holding onto those leaves a little longer. We're maturing a little longer. We're putting more moisture into those pods. We're increasing that test weight. So we've got a healthier plant that's holding on a little longer, because we went at this location, I believe it was 60 days without a drop of rain. So very tough, hard red clay in northern Alabama. And you can see we had a 3% yield uplift, 2 bushels. And again, we're seeing those higher 70% wind rates.
Okay, so I'll switch gears here a little bit, back to wheat. Wheat was the first product that we had as far as indigo seed treatment back in 2017, and so we've been doing a lot of work with different wheat products over the years, and it's obviously grown in an area of the U.S. that oftentimes sees some pretty significant heat and drought stress. So we've had, like I said, had some really good success. I'm going to focus this morning on W13, which has been a product we've had around for several years, and kind of show you here. This is this last growing season on the right, some yield improvements we saw with our biotrinsic with this W13 product across the state shaded here. So this is just a plot that we had out, or that I had out in near, once again, in southeast Kansas near Elk Falls. But typically what we see on wheat is we'll see, early on, a lot of times we'll see differences in early root development here, as you can see in this January 31st picture. We just have more fibrous roots and more of them generally. And then also what I typically see is a better early initiation on tillering. And you can especially see this in the April picture here down, below. Those plants, the treated, have at least 3 tillers per plant. The untreated, it's more typical to find 2 to 3, and so, we're carrying that improved root biomass through as well, going into the spring. And the left here, this just shows an as planted map of this field, showing you, we had the same variety. The only difference being the indigo seed treatment there in the blue. So one thing, this field had quite a bit of variability. We had kind of a steeper hill on the right side there. This is a harvest map showing kind of overlaid with the planting map, and so you can see the variability that we had. We had a low area there. If you can kind of look, kind of in the middle of the field where the purple is, there was a low area where the yield was off as well. But what we're able to do in these fields that have quite a bit of variability is do yield analysis. And so, through My John Deere or Climate, or, there's other software out there that you can do this as well with. But, what I did is just basically selected a 1 acre zone on each side of the treatment area, the split. And, as you can see here, we had a really nice gain. We had a 7.4 bushel advantage. If we looked at this field as a whole, there on the, in the yield column on the right, you would actually think, that the untreated was better just because of, the differences in soil conditions, slope, and then, also there's a creek on the right side of this field that was, has better soil next to it as well. So this is this basically, I did this on several fields this last year and saw really good results, pretty much on every one of them. I wanted to show this example just because of the differences we saw in growing conditions between the 2021 harvest year and 2022 harvest year this year. 2021, we had quite a bit of moisture in the spring. We had a period of almost 2 weeks where I'd say our soils were pretty much completely saturated. And as you all probably know, wheat's not a crop that likes wet feet. So it did impact our yields pretty substantially, especially in southeast Kansas. But this was a trial I had out where we had, a same variety again with the treatment kind of in the middle of the field and the untreated on both sides. So one thing that I thought was pretty interesting, this product is the same one that we've been showing in the other slides. It's a product that's, primarily for heat and drought stress is how we sell it. But what we have seen in these years where we have excessively wet conditions is, that better plant health early on in the season, better early root development, allows those plants to handle those excessively wet conditions better. And you can see here, if you look in this left picture, there's a difference in color and of plant height in this low area across the field. And once again, I think we're seeing because of our improved root development, we're able to pull in more nitrogen, more sulfur, which is allowing those plants to be healthier, and with those wetter conditions that we experience, those saturated soil conditions. So going to harvest, you can pretty much see here on the harvest map to the line where that, we can see more greens and yellows in the area, the treated area of the field versus the untreated area of the field. And, we ended up having a 6.3 bushel advantage, on this field. So, I guess the thing is, when we're out talking to growers about these products, we focus them on, certain stressors, but a lot of times having that, that strong foundation out there with that better early growth, that better early root development is going to help, in times maybe, like I said, of excessive moisture, as well as excessive heat and drought stress too. So it just is a testament to, to how these products work, when the plants are subjected to a number of stresses.
Okay, I don't know how many of you on here grow cotton, but if there are some cotton growers, we have one product for cotton that is WT29. The way I kind of talk about that is, of course, cotton is grown in hot, dry areas. So just think of West Texas if you want to think of the kind of area it goes in with hot or heat and drought. So again, we talk about those stresses that, especially cotton is a tree that we grow as a perennial. And so if you think about a tree, the first couple of years of that tree growing, it really builds that root system and starts building the house for the plant to start setting fruit. So if we get that stress during that early vegetative pre-bloom window, we're seeing an 88% win rate with a 34-pound lint per acre uplift. If that heat stress comes during bloom to bowl set, then we're seeing a 26-pound of lint uplift with a 100% win rate. And just heat stress just seems to be something that comes with cotton no matter what. So like Matt alluded to, that we used to place these products in those stress type environments. But what we're seeing a reaction, or not a reaction, but a response to having these microbes inside of those plants, no matter the environment that they're in. So heat stress is one thing that we have all the time in cotton growing areas, and we're still seeing a 86% win rate. This is a field that I had in 21, and this was in West Tennessee. And you can see the treated is on the right, the line that shows the untreated areas. And this was about a week to 10 days after defoliation. So we sprayed a defoliant plus ethifon, which is a hormone, to open up those not quite mature bolls yet. And as you see on the right, we have a majority of those, that top crop, open, as opposed to the untreated, where we still have some hard lock bowls, some that aren't going to open because they're just immature and don't have enough cotton to open up and let us actually get to it. So we had a more mature plant in the treated as opposed to the untreated. This is a picture from 2021, also in northern Alabama along that Highway 72 corridor, that we split the planter, and this was taken in July, I believe. And you can see the striping of the planter all the way across the field. And then over on the right is a yield map that also lines up with the same striping from the planter. And as you can see, there was a 150-pound increase just in this field alone. So I know this may look kind of busy here, but the picture on the left is from 2021, and the other two is from 22 that was harvested about 2 weeks ago. So the picture on the left from 21, you can see the black line. To the left of that black line was treated. To the right was untreated. And there was a 135-pound increase in the treated as opposed to the untreated. Well, after we harvested, the grower got to telling me that he actually had us in the better part of the field, that about 2/3 from the top of that picture down, there's a rise in that hill. I mean, there's a rise to a hill in that field. And so I went back this year with the same product and said, okay, let's put it in the lesser yielding part of the field. And again, with the drought that we had this year, 50 plus days of no rain in this location, and the treated was in the lesser yielding part of the field, there was still a 23-pound increase with the treated over the untreated. Again, we're seeing the same story in cotton as we are with the other products as far as a bigger root mass. We're getting more lateral roots. We're getting more root hairs to explore more soil. We're also being able to take up more nutrients by doing that. The picture at the bottom on the left with those cotton bowls and those jugs, that's treated and untreated, and those are all first position bowls from a plot down in central Mississippi from 2 years ago. And what we're seeing is we were having more first position bowls. We were holding on to those longer throughout the growing season. Of course, that's where the money is. That's the bolls that mature the longest on the plant are the first to be set, and that's where all the money is. There's another picture of some young cotton plants. This is probably right at squaring, and you can see that the treated is at least 18% greater biomass, if not more. And you see the same things in the seedlings in the lab. So I guess that concludes the presentation.
All right. We have a number of questions from the audience. I'm going to start working through them here. Remember, guys, if you have questions, type them into the Q&A bar, and we'll try to get to as many as we can before the end of the webinar today. One question jumped out at me, and I think it speaks to our audience probably more directly than some of the other questions. There's a question from Bob Wayland. Do these organisms build up in the soil or tend to accumulate over multiple years if no-till is used? And I think that question speaks to number one, do you guys see any kind of variability in the frequency of required application for the best result dependent on tillage practices? And then number two, have you noticed any difference at all as far as tillage, conventional tillage goes?
I can tackle this one first. So as far as tillage practices go, really we haven't seen a huge difference, I guess. I have a number of growers that I've worked with for several years that have used our products, both in reduced till as well as no-till situations, and we tend to see improvements under those conditions as well. What was the rest of that question, Brian?
The question itself was, do we see accumulations of these organisms in soil over time?
So typically, no. We won't. They don't reside in the soil for that long of periods of time. We do see benefits in soil health, from reduced tillage, and but our products really aren't designed to live in the soil for that that length of periods, I guess I would say, from one crop to the next.
Okay, our next question is from Paul Gromberg. He asks, what microbe placement gives the best opportunity to get an increase in yields from a seed treatment? Anybody want to try and answer that?
I don't know. Is that PR?
I'll take that. What microbe placement? That we have data that supports stress factors. We also have the past couple of years kind of moved out of that frame of staying on that stress acre and covering all acres. So I guess really what are you after? Is it water? Is it nitrogen? Because especially in corn, we talked about W12, which is a water use efficiency product. You're getting other benefits as a larger root mass to take up water, take up more nutrients. You're exploring more soil. We have another product that I have had great success down in the South. I don't think it's had the best success kind of up in Matt's area, but it's M3334 is the name of the product. And it actually has a microbe that can fix bacteria or nitrogen, can mine nitrogen for organic nitrogen and make it more available to the plant. So I guess we need to have a conversation of what are you really looking for, because we can target water use efficiency or nutrient use efficiency, but we're not just targeting the stress acre. We're pretty much going broad spectrum now, because we know these plants or these microbes survive inside the plant. Like the other question about, do they build up in soil? No, they build up inside the plant. They colonize the plant is where they go. They don't stay in the soil.
Okay, our next question is from Lisa Blazer. She asks, are the seed treatments crop specific? And I know you tackled that specifically in the number of products that you were speaking about. But have you identified certain microbe genre that are only associated with certain crops? And I think that's a little bit more broad-based. I think Mechdam might have the answer for this one.
Well, I know when we do the isolations and when we get the samples and we isolate the microbe, we know exactly where it comes from in terms of the tissue, like is it coming from the root or leaves or the rhizosphere. But in terms of the product placement when it goes into the field, they like once they put it in stage 2, I know we have labs here that does stage 2 screening into the microbes. They go specifically into certain crops. If it has a genome that says that it has an ability to fix nitrogen, they would probably try to put it in a nitrogen stressed plant. And I think currently, and they probably try it in soy or corn and see if it has that effect, and it moves on through the pipeline and they see which crop specifically works in and what stress it works in. And then finally they decide to when they put it into the field, they will know which crop and what stress it works towards.
Okay. This next question is probably best for Kevin. It comes from an anonymous attendee who asks, as a quality control measure, do you sequence the finished product against the original microbial isolates destined to be in the finished product? Do you guys run the genome sequencing on the original culture versus what goes out to farmers, I guess is the...
Yes, most certainly. I mean, that is one of the requirements on each specification or for every one of our products, is that we sequence the final product and confirm that it matches from a genetic sequence, the microbe that we say is going into there. In addition, we make sure that there's no other microbial species or fungal species in our products. We have a contamination specification, and that the products that you purchase are 99+% the species that's listed on the product. There's not additional bacterial fungal species in there. So.
All right. Our next question is from Mark Jansen, and I think Matt and PR might have the inside track on this one. Have you guys done any trials on long-term organic fields that historically use legumes, manure, livestock, etc.?
I can answer first and let PR jump in. I have not. Like I said, I've got growers that have been, and doing soil health practices such as reduced tillage, using cover crops. But as far as going completely organic, I haven't. I don't have anybody that's actually doing that. But like I said, I know from what I've seen with how our products are performing with those growers on that are improving soil health. I would say that our products would continue to perform well in those conditions. But I don't know, PR, have you seen anything specific to organic situations?
No, not specific. I mean, I think we're trying to get into that, to do some stuff. I know we're doing some long-term studies over adding livestock into the row crop system and how that truly affects the organic matter and all the soil health practices that livestock can bring to a soil. So, but no, we haven't done any testing in those conditions with our microbes as far as I know.
All right, and this next question is probably for the whole group, depending on expertise. It comes from Jim Guy, and he asks, how much nitrogen is it supplying to the plant?
I don't know, Matt or PR. So I think, with our nitrogen use products, right, we aim to better utilize nitrogen that is available in the field in terms of reduced nitrogen inputs and things. I don't know, PR or Matt, if you can comment more on, what we recommend there.
Yeah,
So,
go ahead, PR. All right, I'll jump in first. So PR alluded to the fact that we do have the one product, the M33, M34, that does help supply some additional nitrogen in our testing. We've seen that. We have not come out, I guess, and said that we're wanting to reduce nitrogen rates because of our products. The way I look at it is, these products are improving root development, early season root development, and because of that, we are able to absorb more nutrients in through the roots. throughout the growing season, and I think we have more nutrient availability. Well, I know we have more nutrient availability because of that. We are doing some testing, I know, in some trials with some nutrient use products that will be designed specifically, in that arena, I guess, as far as supplying organic nitrogen. And then we'll probably be looking at. I know we've been doing some testing with that as far as looking at reducing nitrogen rates with those products, but we don't have those commercially available yet.
All right.
Anything?
You took the words out of my mouth, Matt.
Okay.
All right. Our next question is from Jeff. It could be either Quiter, Keeter, or Quitter, I guess. If we're using a fungicide seed treatment, how would you apply your guys' products most effectively?
I'll take this one. We've got several different options, especially with Kevin talking about the 3 different formulations that we have. Like, we're trying to broaden that window of how you can apply it. So your retailer, dealt with some retailers that are perfectly okay with adding another tank into that process of treating the seed, and then our product goes into that tank just like the rest of the treatments that are there. We also have the flowable powder that can go on all the way up to the hopper box of the planter. So we have a broad window from treater all the way up to the planter box, the hopper box on the planter. If you can't get the retailer to do it, but we have a broad window. So effectively, just like Kevin talked about, we know that even if you put it in the hopper box with that scoop, we know that it's getting down through all of those seeds in that hopper box, just like talc wood or graphite wood that you sprinkle on the top as you take off the plant.
All right. I know you guys had said you're not looking to replace nitrogen. This is a question, I guess, that speaks to general practice from some of the trials. It's from Cheryl Castine. In general, have you found that farmers using your products use less fertilizers? And is there a difference? And I know you guys didn't speak to organic farming much, but have you seen a difference in fertilizer use among your trials?
I'll jump in. Specifically, we're not, like I said earlier, we're not recommending, nutrient reductions because of our products, but the way our products work inherently, with improving root development, I do feel like and know that we have more nutrient availability, and a lot of times that's why we see color differences out there, and better early growth is because we have, we're bringing in more nutrients. We have it's we've got more pipes to bring in more nutrients to that plant. It's kind of what it comes down to.
Okay. The next question is from Brooks Bren. Specifically about the soybean products. Can your soybean biological with inoculant be applied in furrow with a micro applicator similar to how some liquid insecticides are applied? And the idea is she understands not to, or he understands not to apply it with an unclean applicator. If it could be applied this way, if there could be an interest in dry or low organic matter soils.
I'll take a pass at that one then. So our products at this point are intended to be applied as seed treatments. From the kind of their physical characteristics, it would seem that you could apply them in furrow with some of the liquid technology used to apply in furrow. But one thing you have to consider is that when you apply as a seed treatment, we're putting those microbes directly on the seed. And then when that seed is planted, they're immediately adjacent to the seed so they can interact and colonize that plant. Right there. When you apply in furrow, again, depending upon the equipment being used, the microbes may be more spread out, dispersed. They're not always directly next to the seed, and so your dosing can be different. So that's where our products currently are formulated, and the concentration of the microbes are intended to be for seed treatment. They're not really intended to be for in furrow at this time. But theoretically, there could be some additional product development and changes in the product, particularly around dosing, to allow for in furrow. But at this time, it's focused on seed treatment.
We have several questions that are kind of in a similar vein. One from an anonymous attendee and I believe you mentioned this just briefly at the end, and I was hoping I could get you to elaborate. Will the return on investment be greater in stress conditions? And then another question from Gord Specksnyder. Most of your real world examples show response under stress situations. Is it fair to say that we can expect less response in good years? And by extension, does it make sense to concentrate use of these products on poor fields, i.e. the fields most likely to have crop stress?
I can jump in here. So, I would say overall, probably yes. I mean, from the data that we've got in the last several years, when we do see stress conditions, we typically do see a better response. But with that being said, like to PR's point with the M33, M34 product on corn, that product has been one that we have seen, probably perform a little better under more ideal conditions, where we don't see that stress. So, like I said, we have different products that fit in different situations. With the product on wheat that I pointed out with the W13, that product has performed well in good conditions as well as stressful conditions. We have a product WN29 on wheat that I would say performs better in hot, dry conditions. We tend to place that product more in the the Western High Plains part of our region. So it really goes back to the product, I think. And certain products do perform better under stress, but I think we have the products there that will perform well under more ideal conditions, as well.
And then we have two questions that are follow-ups to the earlier question about fungicide. One, will seed treatment fungicides kill the microbes in your product? And another similar question, can they please clarify the fungicide seed treatment answer? Is there a fungicide seed treatment? If there is a fungicide seed treatment, won't that kill the fungus species in the applied product and prohibit root colonization?
Yeah, I'll jump on this one too. So again, depending upon the product that's being used, they have different compatibilities with different chemical pesticides, particularly in this question, the fungicides. So one, I always refer you to the product label itself, and then working with our agronomists and account managers to look at the product you have, the fungicide you may be considering using, and how best to use those two products together. So as PR talked about previously, some of our microbial products, some of those liquid products, they are very robust and they can be directly tank mixed with your fungicide and applied to seed, and there's no issue with compatibility. Some of our other microbes are a little bit more sensitive, and if you read the labels on them, they can't be tank mixed and they have to be applied to the seed, post seed treatment of the chemical pesticide. And so, again, as I mentioned during my portion of the webinar here, those microbes that are a little bit more sensitive, we tend to formulate those as powders, flowable powders. And so that's where once you apply your liquid pesticide seed treatment, then you can just dust on that powder on top. And the reason you don't have compatibility issues then is that fungicide has gone on, it's dried onto the seed, and now our powders are going on then dry too. And so the interactions between the microbe and the actual chemical fungicide are really non-existent because everything in its nice dry state, there's no liquid interactions. So hopefully that provides a little bit more clarity there. Again, it really depends on the individual product, the microbe you have, and following the label to see if you can, go the very straightforward route, mix everything together and put on for the robust microbes, versus maybe you're working with a microbe that's a little bit more sensitive and that's a powder and you have to dust it on after you apply your chemical fungicide.
That's maybe the biological version of the answer label is law, right, for chemistry products?
Sure, yep.
All right. The next question, it speaks to my heart, is a short-term resident of the Seattle metro area. This is from Cheryl Castine. Have there been any similar testing of microbes in the Pacific Northwest for your guys specifically? And if you guys don't have a presence in the Midwest, is there another company that they can approach?
Matt, PR, you have any insight there? I mean, I know a little bit of history of looking at some wheat in the Pacific Northwest, but in terms of current activity.
Current activity, I think Colorado is about as far west and north as we get out there. Matt, do you know of anything past that?
I don't. Yeah, I know we've got Andrew out there that's promote our products out there in Colorado, the Front Range primarily. But yeah, I don't know that we're going out to the Pacific Northwest at all. Maybe that's something we need to look at in the future for sure.
I can jump in this one at least. I know when we did collections, we also collected from New Mexico because there was a partnership with a farmer in New Mexico. So we do have microbes that were isolated from farms from New Mexico, so further west.
All right. The next two questions are from Darcy Nestabo, and we do have a large contingent of no-tillers that are in Canada, where it's a very widespread practice. So this question is: do you have registration for Canada? And a similar. Well, I'll let you answer that one first, I think.
So again, it's going to be product specific. At this time, I believe we have one product that is registered in Canada, but not all of our products are registered in Canada. So again, depending upon your crop, the environment, your agronomic practices and things, that one product may be appropriate or not. I think it would have to be a follow-up conversation to see, if there's a match there. But definitely always interested to hear about, what your requirements are in considering future products.
And the next question, also from Darcy: have you guys done any work with brassicas?
Megdum, do you know off the top of your head in looking at that genre? And so it looks like we may have several brassica species in the library, but we don't have any commercial products at this time under that genre.
And then this one relates to, I think probably also Matt and PR. Will a biological product work better in a sandy, lower organic matter soil compared to heavier clays?
I'll let you tackle that one, PR.
Will it work better in a sandy, low organic matter than heavy clays? I've had it in both. And I don't know if it works better because I really can't compare them because they're not usually close enough that I can compare even the same weather pattern. Ooh, that's hard to say. But again, I don't know if it'd work better, but I would definitely go after one of the water usage products in that sandy, low organic matter area. And I guess it depends on what crop you're looking at in that area too. But I've seen it perform in both areas, but I don't know if it works better in one than the other.
Okay, and these last two questions are probably going to be for Kevin. Another question from Darcy. He says he's making the assumption that your inoculants are bacteria dominant, and I mentioned during the stabilization product sometime, or one of the products of manufacturing, guys sometimes add additives like mycorrhizal ingredients. So how do we address the fungal component?
So it's true that the majority of our products are bacterial, but we do have commercial products that are fungal. Megdum, correct me here. I think, within our internal library of products in the pipeline, it's roughly 2/3 bacterial, 1/3 fungal. Is that roughly it, Megdum, or are we?
Overall, it's even less fungal. Okay. I would say even less than that.
Okay, maybe it's more a 75-25 split, but we do have commercial products, both that are bacterial and fungal. I think the one WT29 product that PR was highlighting in cotton, that's a fungal species. And sorry, what was the second part of the question there, Matt?
Well, hang on, let me find it again. Oh boy, I think I lost it. Essentially, the question was, how do you integrate the fungal component into your treatments? And I guess you guys seem to have answered that. So, but the final question.
I mean, again, sorry. Any fungal organism that we turn into a product goes through the same development pathways. And again, depending upon the physiology, how well we can keep that fungal species in a dormant, preferred a sporulated type state. It then dictates the type of products. I will say that most of our fungal products are flowable powders, although we do have some that are soon to be released as a liquid, a water dispersion type product. And then, of course, that will dictate how you use those fungal products and treat seed with them, whether you can actually mix them and apply them with liquid seed treaters or if it's dusted on as a flowable powder.
Last question is from Lita Lorano, and I assume this is a QC question. The question is simply, how about viability tests? I think that's a follow-up to the sequencing question from before. You guys do viability before and after?
Yep. So that too is people are picking know our specifications, and you can imagine what it looks like. So yeah, one of the first things that we measure when a product is manufactured is we measure what's called the titer, or the concentration of the microorganism in that product, and confirm that it meets the target titer at the time of manufacturing. And then, of course, we have an expiration date on the product itself. And so that through that time period, as a product sitting on the shelf at our recommended storage conditions, you're going to maintain that product titer, that product concentration, so that when you go and treat the seed, you're going to get the correct dosing. So that, along with confirming the identity of the species and that it's pure, are kind of the three main metrics through our quality assurance process. And of course, we have additional metrics too around kind of more physical chemical characteristics.
All right. That's all the questions that were in the queue. I think we're about done here. So before we conclude, just a heads up that when you log off, you'll receive a questionnaire to complete. We'd really appreciate it if you fill that out and submit it, because it'll help us plan for future webinars. Second, we'll be making an email, or we'll be making available via email to you a replay of today's webinar in the event that you'd like to go back and review the presentation. You'll also find that replay up on our No-Till Farmer website shortly. I can say from experience, typically 24 to 48 hours. We like to cut some of the stuff in the beginning where it's me trying to figure out how Zoom works, but for the most part, it's a pretty good time. So once again, thank you, Mectum, thank you, Kevin, thank you, Matt, and thank you, PR, for leading this discussion. Hope everybody got some good ideas for managing biologicals and ideas for products. And of course, thanks to all our farmers for joining us. Goodbye for now, and have a great day, everybody.